Yes, so hi, I'm Savannah, a research fellow at Loughborough University, but I thought I'd just give a little bit of a background of how I ended up where I am now. For any ECRs on the call or anyone watching this, I've had a somewhat non-traditional route towards a sort of research position. And also working across stable isotopes across different disciplines, which will sort of preface what I'm going to talk about today. I did my PhD at the University of Nottingham, and I was working in paleoceanography. So I was using compound-specific stable isotopes on diatoms and foraminifera, as well as bulk sediment stable isotopes, to look at the biogeochemistry and interaction of productivity and nutrient cycling in our oceans, in particular the subarctic North Pacific, and how that varied over the last million years. I then moved into a non-academic role as an aquatic environmental consultant, so not stable isotopes at all, but this gave me some skills in sort of aquatics and particularly contemporary studies, and this is sort of where I'm working now. The main experience I have that sort of brought me to this talk is that I worked as a stable isotope research technician at the British Geological Survey for a couple of years. And during that time, I was responsible for the preparation and analysis of a number of samples for carbon, nitrogen, and oxygen stable isotope analysis, including running the mass spectrometers they have there. After that, I moved into some teaching positions. So I then taught at Nottingham Trent and the University of Nottingham in sort of across ecology, environmental science, physical geography. But throughout those roles, I was really sort of embedding stable isotopes as part of all of these different themes of teaching. And that's ultimately led me to my current position at Loughborough, where I'm now developing stable isotopes for pollution tracing, and in particular understanding the impact that sewage has on algal blooms in standing waters. So I'll be covering a little bit of a specific case study I've been working on towards the end of this talk. So in terms of what I'm going to talk about in a skills and training perspective is that I'm going to start off with stable isotope fundamentals. So what is a stable isotope? Why do we want to measure them? How do we go about it? And what could we measure? And then giving some key examples from traditional stable isotopes. So there's a series of light isotopes that are very well used in academic and non-academic research. But I'm going to cover some of the key examples that I've come across from my own research. I'm then going to move into sort of more specifically around aquatic biogeochemistry, which is really my own sort of specialty. I'm going to talk a little bit about that pollution tracing and how we can use those, and cover a couple of case studies that I think demonstrate this quite well, at least with some nice figures. And then I'm going to move into talking about some of the work that I've been doing to develop phosphate oxygen isotopes, which is not a more novel isotope system that we can use to trace phosphorus cycling in the environment, which is quite important for sewage, and talking about that case study I've been doing at my study site, which is the Rutland Water Nature Reserve. So first up, what is a stable isotope? So stable isotopes are essentially just different forms of the same element. So we have in this figure here the two stable isotopes of carbon, which are carbon-12 and carbon-13. There is also a radioactive isotope, which is carbon-14, that people will be familiar with from a sort of dating perspective. But when we talk about stable isotopes, we're not talking about those which decay. What you'll see from the carbon-12 and carbon-13 is that they have the same number of protons, but they have a different number of neutrons, which means that their atomic masses are different. And there are different abundances, natural abundances of these two different isotopes within the environment. So we know that carbon-12 is our most dominant form of carbon in the environment. About 98.9% of carbon is a carbon-12 molecule. However, we also have some carbon-13 at about 1%. And it's because of this slight differences in the isotopes that on the periodic table, we see the atomic mass as 12.011, which is a weighted average between the carbon-12 and carbon-13. And as I say, we're talking here about the stable isotopes. The traditional light stable isotopes that have been measured for a very long time include oxygen, hydrogen, carbon, nitrogen, and sulfur. But there are a wide range of other stable isotopes that are increasingly being measured, including non-traditional isotope systems and metallic elements, which are highlighted in this periodic table on the right. I'm going to be focusing this talk around the traditional stable isotopes, as that's where my experience really is, and they have a key role within biogeochemical cycling. The principle of how we use isotopes is that because of their mass difference, when they are used in biogeochemical processes, there will be a discrimination between how much of each isotope is used, and we call this process fractionation. A key example of this is, for example, if an organism wants to take up some carbon to use it in their cell, they will use up a larger proportion of the lighter 12 isotope than its heavier counterpart because it requires less energy to move that carbon 12 molecule. It's because of this difference in speed and energy between these two isotopes that every biogeochemical process has a different rate of fractionation depending on what that process is. And we can use this principle then to track stable isotopes through the environment to understand what processes they've gone through. The process of fractionation is a very common principle, and one that's really well established and easy to follow is within the hydrological cycle, where we have oxygen within H2O molecules. There's 3 isotopes of oxygen, but we tend to focus on oxygen 16 and oxygen 18, which are the 2 most abundant. We find that the lighter isotope of 16O we know requires less energy to use in a number of processes than its heavier counterpart. So within the hydrological cycle, what this means is that evaporation, which is an energy when it requires energy to sort of transform it from a liquid to a gaseous phase, that happens more quickly for our lighter 16O than our heavier 18O. So evaporation has a negative fractionation effect where you have lighter isotopes in the clouds that form from that evaporation. Alternatively, then, when those clouds move inland and we have a condensation process, which is an energy loss process, the heavier 18O is going to be rained out more quickly than its lighter isotope. Essentially, what this means is that you could measure the oxygen isotope of a water molecule anywhere within the hydrological cycle, and it will have a distinct isotopic signature that depends on what processes have occurred. The idea here is that, for example, if you measured rainfall that was further inland, it would have a lighter isotopic signature compared to that at the coast, where the heavier isotopes have been deposited first. And we see this as a principle then in archaeology, because where we have drinking water supplies that are either more coastal or more inland, our bones and our teeth will have oxygen isotope signatures that match the drinking water supply of where we've been born or where we've grown up. The way that we express and talk about isotopes is using what we call a delta value, a delta notation. This is because really what we're measuring is the ratio between 2 different isotopes and how that changes over time. And we always measure this relative to a standard, and that standard will change for each isotope system. Because the differences that we're trying to measure here are pretty small, we express them as parts per thousand or per mil, which is denoted by this sort of percentage sign with the extra 0 on it. So here we have the delta 18O expressed per mil, which is calculated by the ratio of oxygen 18 to 16 within your measured sample, in comparison to the standard times by 1,000. If we were to measure our oxygen isotope value as plus 10, the way that we would talk about this is we would say this sample is enriched in 18O relative to the standard by 10 parts in 1,000. Essentially, it's isotopically heavy and it contains more 18O than our standard value. Similarly, if we have a negative value of negative 10 per mil, we would say that the sample is depleted in 18O relative to the standard by 10 parts in 1,000. This is isotopically lighter than our standard value. It has less of that heavy 18O within it. So how do we measure stable isotopes? The easiest way of doing this is that we'll convert our elements into a gaseous form. So there's numbers of ways of doing this. It could be through combusting it with heat, particularly if we want super high temperatures, we'll use pyrolysis. You could react, for example, if you have a carbonate, you would react it with an acid to produce CO2. Or in the case of nitrogen, you can also use bacterial transformation, where you take it from in solution to a gaseous form. Once it's in a gaseous form, we analyze it using an isotope ratio mass spectrometer, which is shown here in the diagram on the right. The way that this works is your gas will pass through the machine and it will ionize that gas, exciting the ions within your system. The heavier isotopes will be excited less than the lighter isotopes, and they will travel slower as they move through the magnet, as you can see in this figure. The difference in travel, because of this difference in mass as a result of these isotopes, is then detectable by our ion detectors at the end of our mass spectrometers, where they then measure the number of ions hitting that detector to tell you how much of each isotope is within your sample. And as I say, these are the traditional stable isotopes. So we have oxygen, hydrogen, carbon, sulfur, and nitrogen. Each one of these has different numbers of stable isotopes, but we're always comparing the two that are the most abundant. So for oxygen, this will be 16 and 18. You'll also see that there are a range of standards that we can use to compare against, even within the same isotope system. For example, for the oxygen isotope system, we have different standards depending on what material you're analysing. So for VSMO, which is the Vienna Standard Mean Ocean Water, this would be our water standard. The Vienna PD belemnite, which is used for our carbonates and carbonate standards. And then the oxygen with the air, which is used for gaseous comparisons. Each of these systems has different standards, so it's really important that if you're ever running your own samples or comparing them against other people's data, is that your standards are comparable. And what can we measure? Well, really almost anything. So some of the key examples are outlined in this table on the right from Carol Kendall's paper. So you can read this in your own time if you'd like to. But as a brief summary, we can measure in particular things like the water. We can measure waters in surface waters, groundwaters, lakes, oceans, rivers, pore waters, waters within cells. It can be really anything. Soils and sediments are also very commonly analysed, particularly in biogeochemical studies where they represent the processes going on in terms of biogeochemistry within a system. For example, at the bottom of a lake, bottom of the ocean, or within riparian habitats. You can also measure rocks and geological materials. This will require a level of processing, pre-processing in terms of grinding and homogenizing that sample. Plants and algae are also very commonly analysed. You can do this either as a bulk sample by grinding it all up together, or increasingly people are using compound-specific isotopes. So, for example, measuring the hydrogen isotopes on leaf wax is very common, and looking at root structures and different types of the plant. Biota are also commonly analysed, fish in particular. You can look at the gut contents of fish to understand what their primary sources of food are, and think about trophic structure and food chains. You can also measure gas itself. So instead of converting it to a gas, you can take an atmospheric sample. For example, you could use this with a gas sampler if you're looking at sort of CO2 or methane flux, or if you're taking you can have sort of extractions of gas from sediment cores. And then fossils and archaeological remains, which are certainly very prominent in a paleo perspective. So things like the fossils of zooplankton and algal groups are very common, but also archaeological remains, things like bones and teeth from humans and others. So a few classical examples. As I mentioned, the hydrological cycle and oxygen isotopes are well studied, and there's this principle that light isotopes are preferentially evaporated over their heavier counterparts. And this is a really key fundamental principle for how we approach paleo-environmental reconstruction. Because we have these in the past glacial periods where we had large amounts of water that were locked up as ice sheets on the land, what we know is that as we have that isotopically light water that is evaporated and moved inland and rained out onto the land, that then freezes and locks up. So our glaciers will be enriched with isotopically light oxygen. This leaves more of our heavy 18O within the seawater behind it. And that seawater oxygen signature is then preserved within the carbonate shells of organisms that live within the ocean at that time. So we end up with this shift between different isotope ratios of 16 to 18 as a result of the size of glaciers on the land. And this is really what underpins a lot of paleoceanography. So here we have the LRO4 stack. This is a combined record of a number of different ocean sediment cores where they've measured the oxygen 18 of foraminiferal shells within those sediments over the last 1.8 million years. These shells preserve those oxygen isotope signatures of the ocean water, which is a reflection of that balance of precipitation and evaporation on glacial interglacial timescales. And we use this LRO4 stack commonly as part of our age model process. So if you take a new ocean sediment core, this global benthic stack is often used to tie to understand what age of sediment you're looking at. So it's a really important basis for how we know how old our sediments are. For carbon isotopes, we're looking more about what carbon cycle is, and in particular, sources of organic matter within the system. We know that biogeochemical processes tend to require less energy and use lighter C12 over C13 as a classic example. But actually, depending on what type of plant you are and the types of CO2 transport systems you will use different ratios of these carbon isotopes. And we can therefore use this to trace what sorts of plants or organic matter is growing at different times. There's clear distinctions between C3 and C4 plants in particular, but also a distinction with plankton and autochthonous producers that we find in our oceans and lakes. These fractionation processes that each type of plant or system, each type of plant or algae is using, is then preserved within the organic matter that falls into the soil or into the sediments at the bottom of lakes and oceans. And it's on this principle that we can then reconstruct past vegetation changes. Nitrogen isotopes, however, are not used for that. They're used more commonly to understand things about the nitrogen cycle, and in particular, utilization of nutrients and how they are cycled. And this really is because nitrogen is limited in many environments, particularly the ocean. So instead of representing sort of a distinct source, often the nitrogen isotopes will reflect the amount of nitrogen that's being used relative to the amount of total productivity. This all stems from the basis that, as before, organisms will use a lighter, preferentially use a greater proportion of the lighter 14N. And as they use up a larger proportion of the 14N, they'll have to start using a little bit more of the 15N as a proportion. So when we measure our nitrogen isotopes, the heavier the signature, i.e. the more 15N that's incorporated, the greater the proportion of utilization is relative to the pool of nitrogen to begin with. To outline this is a simple schematic. If we measured our isotope value of 6, here we have a grey circle which represents our nitrogen pool and little cells that are using up those nitrogen. If we increase the productivity but keep the pool of nitrogen the same size, what this will do will cause an enrichment of our nitrogen isotopes. If, however, we keep the productivity the same, but give lots more nitrogen to the system, what we'll see is a decrease in that isotope, where less of the heavy isotope is being incorporated into the organic matter. If we make the pool of nitrogen smaller and increase the productivity, we see an even larger increase in our nitrogen isotopes. Essentially, all this is trying to demonstrate that when we have an isotope value, there's an interpretation that's required to understand what else is going on within that biogeochemical cycle to lead to these isotope values. I have no experience with paleo diets, so I'm going to defer to my colleague in this YouTube video if it will work, because another key way that we use particularly carbon and nitrogen is to understand the diets and trophic status of organisms within the environment, and in particular in archaeological contexts. So hopefully this will work.
So, my name is Michelle Alexander. I am a lecturer here at the University of York. I am a specialist in stable isotope analysis to understand the diet of people in the past. When you're looking at isotopes that we use in terms of diet, they're stable. So when you eat something, the signature of that food in terms of its isotopes is retained and stays the same once it reaches your body tissues. You are continuously renewing your bone. So when you eat something, that signature will stay for as long as it takes that bone to completely be replaced. And that usually takes about 10 or so years. Teeth, on the other hand, are a little bit different. They form in sort of layers, if you like, all the way down to the bottom of the root. So if I look at this tooth root, what it will tell me is what someone's diet was like between the time that that tooth was forming. If we're looking at a first molar, that might be between sort of 0 years and sort of 10 years of life. What we're interested in is looking at the protein within bones. To do that, we need to dissolve away the hard part of the bone, so the mineral, and then we filter the protein to make sure that we get just the protein. We weigh it out into tiny tin capsules and we burn it in a mass spectrometer. The burning it just releases it as a gas to measure the isotope ratios. We look at carbon and we look at nitrogen. If you're plotting down here on the graph with a kind of low carbon and low nitrogen, then you're probably eating plant-based diet with some probably animal foods in there. High carbon, high nitrogen puts you over here where you're looking at seafood. If you are eating a bit of seafood and a bit of kind of land animal protein, you'd plot right in the middle. So what we wanted to understand was there a change in diet between childhood, and remember that comes from the tooth, and adulthood, which comes from the bone. All of the triangles plot quite close to their partner crosses. So what we're seeing is diet between childhood and adulthood at these sites in this Brighton area didn't change that much. And one of the things we think is going on in the Roman period is you might be seeing these higher nitrogen values associated with people of higher status, so higher class. And then people with low nitrogen values, these are people who can't afford to have a lot of animal protein. So when we look at this Brighton individual, we can see that they plot not among the people with the least amount of kind of protein in their diet, relatively lowish status, but still with animal protein in their diet. And so by looking at the diet and seeing what people were eating in the past, it can tell us something about society. When you compare them to other people, we can understand that person's place in society and something about the culture of that society as well.
Okay, so that's sort of an overview of some of those classic examples where stable isotopes have been very commonly used for a number of years. I'm going to move slightly towards a specific application now, which is going to be centered around aquatic biogeochemistry, which is my own area of expertise. I'm going to be talking about pollution tracing in aquatic systems, and again about those phosphate oxygen isotopes that are a slightly more novel tracer in this system. So why do we care about aquatic biogeochemistry and in particular nutrient pollution? Nutrient pollution is a pervasive and unresolved water quality issue across the globe, and this has really resulted from the excess runoff of particularly phosphorus and nitrogen into all of our aquatic systems, which leads to eutrophication, which is very high nutrient levels in our systems. When we have high trophic status aquatic systems, we often see symptoms like very large harmful algal blooms, like in the photo here, problems with dissolved oxygen and habitat quality, and drinking water quality issues as well. Traditionally, when we were doing a nutrient pollution assessment, we would be looking at the concentration of those nutrients within a system. So, for example, how much nitrogen is in this water in a milligrams per litre. The problem is when you have those numbers is it's very hard to know where those nutrients are actually coming from. And also, it's very difficult to measure all the different types of nitrogen at a resolution that's high enough to understand the nutrient cycling processes. So we know organisms interact with different forms of nutrients. And actually, this is essential information to underpin the management decisions we need to make within aquatic systems and catchments more generally to prevent future water quality issues. And this is where I think stable isotopes can offer a really great solution. The best way of doing this is using a multi-isotope approach. So much like in the example of the paleo diets, in aquatic biogeochemistry, we tend to use more than one isotope system. And this is really because when we want to know where a nutrient has come from or where the organic matter is from, there are often overlapping signatures. So you can see in the table here, we have the carbon and nitrogen isotope values or typical compositions of different sedimentary organic matter sources. And there is overlap between those. However, we can then plot this on a biplot where we have these different spaces and plot our measured values on top of it to give us an indication of where these nutrients are coming from. So this is a study that has done this. They've taken a range of sediment cores from a lake in southwest China. S1 is in the northern portion, S3 is in the south. And you can see that most of the nutrients here are falling within the range of the soil organic matter. So most of the nutrients and organic matter coming into this lake are from a catchment source. The really useful way of then moving away from this sort of qualitative view with using a biplot is then to do something called end member modeling. So essentially, if we know that the average value and the range of values for each of our different sources, we can plug this into mixed end member models that are often Bayesian frameworks, including SCIR, mixed SCIR, or isosource as examples. And we can then put a number, a quantitative value on the contributions of each of these different sources to these different systems. So this is what's been done for those same cores. And what this confirms is that alloxanous material, particularly again that soil organic matter and C3 plants, are the dominant source of organic matter to this lake. You can also see that S1 has a slightly different signature to S2 and S3, where we have amounts of autochthonous productivity, including plankton and macrophytes, and also the contribution of sewage. And this is because in the northern sector of this lake, it sits quite close to the urban centre. We can also use isotopes to look back in time. So instead of just doing contemporary monitoring, we can actually look at the impact anthropogenic activity has had on lake systems or aquatic systems over time. So these are the sediment core records that were taken from this lake going back to 1870. We have the carbon isotopes and the nitrogen isotopes, and there's a very clear transition in both of these systems at about 1990. And this reflects a period of urbanization within this catchment, where they introduced animal husbandry and aquaculture, as well as wider agricultural practices. Stable isotopes are also used as pollution tracers by looking at waters, not just sediments. So we can measure the dissolved nitrates within our waters for dual nitrogen and oxygen isotopes, so looking at both components of that molecule. We can then, in the same way, plot this on a biplot where we have these different typical end members for each of our sources of nitrate. So you can see in these different boxes, we have atmospheric nitrates, we have nitrogens from fertilizers and precipitates, from soil nitrogen, and then manure and septic waste as well. We also know that every biogeochemical process has different fractionation rates within a system, and the nitrogen cycle is one of our most complex systems. So here we have all the processes within the nitrogen cycle listed by their chemical symbols and the fractionation effect that would have on a given molecule. There are some really distinct ones on here, including denitrification, which has a very strong signature, utilization as well. And we can use this principle to understand how nitrates are being moved through the system, or nitrogen is being moved through a system, by measuring the nitrate isotopes over time. So this was done in this study by Lee et al., which was on a river just south of the Pearl River. It's a tributary. I'm going to pronounce this wrong. I think it's the Zhejiang River. And they measured the nitrates across a transect from upstream to downstream. There are a number of different sources of nitrogen in this catchment, including fertilizer, which is marked particularly by the red parts on this subset map here, but also by urban centres marked by these population densities. This is then the data that they reflect those earlier figures I showed, the traditional figures. So on the left, we have our source apportionment, and on the right, we have our sort of over time thing. We have a dry season and a wet season signal. What we can see is that in the wet season, the nitrate isotopes are sitting more strongly within a soil organic nitrogen space compared to the dry season, where it is slightly shifted to the right, more towards the manure and sewage. We can also see that there's greater nitrate flux happening in the wet season. So we have the nitrate concentrations on the top of our right-hand figure that shows slightly elevated nitrate concentrations. But the isotope signatures actually seem more variable within the dry season. Because of these very small differences between isotopes, it's quite hard to pick this apart. So again, a really valuable thing to do is that end member modeling. And that's what they did here. So we then see on the left our different sections of the river, upstream or mid-lower stream, and then we have dry season on the top, wet season on the bottom. Of course, there is overlap here between each of these things, but the key takeaways is that soil organic nitrogen does indeed contribute quite significantly to the nitrate flux, but actually chemical fertilizer, represented by CF here, is a much larger proportion of the nitrate flux than was indicated from the box plot. We also see that manure and sewage is a little bit higher as a contributor in the dry season, which probably reflects that there's a much larger flux of nitrate coming from the runoff in this monsoonal system. On the right-hand side, they also then looked at just the upstream reach, and they compared the isotopes against nitrate concentrations, or a nitrate to chloride ratio, which is indicative of sort of pollution. What we see is this trend of a negative linear trend, which represents denitrification. So as well as understanding where these sources are coming from, what this paper was able to do is suggest that 12% of the nitrate was being lost in the upper reaches as a result of denitrification in the lower nitrate concentration zones within the upstream reaches. So what about phosphorus? This is more my sort of area where I've been working in the last couple of years. So phosphorus is an essential element for life, and we know it's a key concern within water quality and within eutrophication. The problem we have is that phosphorus has 23 isotopes, but only one of them is stable. So we aren't able to do that ratio comparison, and all of the radioisotopes decay very rapidly, which has been a real stumbling block for understanding P cycling from an isotopic perspective. However, we know that a lot of our phosphorus within the environment is actually bound to oxygen as phosphate, and the PO bond within phosphate molecules is very strong. So what we can do is actually measure the oxygen isotopes within that phosphate molecule as an indicator of phosphorus cycling. The phosphate oxygen isotope system works a little bit differently to some of our more classical sort of carbon and nitrogen systems, because that PO bond is so strong that abiotic processes like dissolution and precipitation, which are key within the phosphorus cycle, they don't have any impact on the PO bond. So they will not change the isotopic signature of the oxygen isotope, of the oxygen in a phosphate molecule. And it's based on this principle that we can then say that different sources have different phosphate oxygen values, regardless of how they've been moved through the system. So we have on the right here a set of sort of, again, traditional ranges of values for different sources of phosphorus and their oxygen isotope signatures. However, it gets really interesting when we think about biotic processes. So intracellular cycling using enzymes can break apart the PO bond. And when this PO bond is broken apart, the oxygen becomes free and can exchange with the surrounding oxygen, which is typically that within the water, either within the cell or outside of the cell. So what we end up having is we have our source phosphate that will have a given isotope value based on where it's come from. Biological enzyme activity will then break apart that PO bond and enable oxygen exchange between the phosphate and the water molecules. The water isotope values are going to be different to those of the source values, say an isotope value of Y. So as that PO bond is broken and the oxygen is exchanged backwards and forwards, what we end up seeing is a shift from the original source isotope value towards the isotope value of the water oxygen. Essentially, this is a biological overprint where we have an equilibrium, eventual equilibrium, where all of the phosphate has been exchanged with water oxygen, and this is a temperature-dependent relationship. And this is the equation here. So if we know the temperature of our water and we know the oxygen isotope ratios of our water molecules, we can then calculate the theoretical equilibrium value for the phosphate where all of the phosphorus has been biologically processed over time. We can then compare our measured phosphate oxygen isotope values to this theoretical equilibrium to evaluate essentially how much of the phosphorus pool has been overturned. If our measured values equals this theoretical equilibrium, it means that the whole of the phosphate pool has been biologically cycled and exchanged with the water. And we're probably therefore in a P-limited system where phosphorus is in high demand. If the measured value doesn't equal this theoretical equilibrium, it means that the phosphorus is not the limiting factor because not all of the phosphorus has been biologically processed, and our isotope value is likely to be a little bit closer to that original source value. And this is what it looks like in data. So this is a phosphate oxygen isotope study of surface waters in a lake that I've been studying at Rutland Water Nature Reserve. This lake receives treated sewage effluent from a nearby water recycling centre. And what we see is on this monthly scale, our phosphate oxygen isotope values in the surface waters typically match our theoretical equilibrium value, which means that for most of the year, phosphorus is a limiting nutrient and is being completely biologically cycled. However, what you can see is in April and September, we have values below our equilibrium, which represents that not all of the phosphorus has had that biological overprint. And happily, these bars also sit very close to our end member value for sewage at this site, which is 15 per mil. We then took this principle and what we were trying to do, I've been doing this in collaboration with colleagues at the British Geological Survey, including Andy Smith and Chris Bengt, who's his PhD student shown in this photo, is then to think about, could we use this principle of phosphate oxygen isotopes to use that as a reconstruction proxy for biological phosphorus utilization in the past? So what we did is we first of all took the standard approach, which is to look at other isotope systems, including nitrogen. This is a sort of plot of that sediment core. At the bottom, we have time. So the lagoon that I'm studying was built in 1979, shown on the left, up to the present day on the right-hand side. Each of the bars across here represents changes in the way that the sewage that is coming in has been treated. And those changes have been as a result of legislation and regulatory changes in the UK that require stringent amounts of phosphorus to be removed from the system. So from 1991 to 2001, they were using what a natural filtration system where the treated sewage effluent was being filtered through this grass plot before it entered the system. But the key change really on this diagram is at 2001 to present, where they've introduced chemical dosing as a treatment for phosphorus. So now they're using ferric sulfate to reduce the amounts of phosphorus within the water. Essentially, what this figure represents is increasingly low phosphorus concentrations over time. However, what we see in our nitrogen isotopes is a significant increase. And what this represents isotopically is that there's been a consistent influx of isotopically heavy nitrogen coming from that treated sewage effluent. We also know at this site that there's been an increase in the total productivity over time, which is shown in the green line on the bottom panel. This is our chlorophyll A. This is a photosynthetic pigment that's typically used for total productivity. So as I mentioned earlier, we know our nitrate isotopes, our nitrogen isotopes are a function of both the amount of nitrogen coming in and how much is being used. And actually, we have such a significant increase in this top panel of 8 per mil. That's really quite a significant increase. We might expect maybe 2 or 3 per mil over these timescales. So what we know this reflects is that it's both an increase in the source of nitrogen coming from the sewage, but also an increase in the utilization of nitrogen at this site. We also know from our photosynthetic pigment record that the predominant change here is that there's been an increase in the amount of cyanobacteria at this site. Cyanobacteria is a nuisance form of algae for many aquatic water bodies because it produces toxins and has implications for water quality. In the blue, we have one type of that pigment called camptoxanthin, which is a nitrogen-fixing cyanobacteria. So it's possible that this large source of nitrogen is coming from all of these different combined sources. We then have at the very top of the core, you'll see a plateau in our nitrogen isotopes. What we think this reflects is where nitrogen has become in excess and the productivity is being limited by something else, and probably phosphorus. So if no more of the nitrogen pool can be used, we enter into a system of P limitation. And what this has ended up with is an increase in another type of photosynthetic pigment called myxoxanthophyll. And this is a pigment associated with a particularly toxic strain of cyanobacteria at this site, which is microcystis for anyone that knows their algae. So what we wanted to do is try and set out and prove that this phosphorus limitation had happened, and could we use phosphate oxygen isotopes to prove this had happened? So what we did is we measured the phosphate oxygen isotopes on a specific pool of phosphorus within the sediment, which we call HCl extractable P. This is the phosphorus that's typically bound to calcium. And the reason we chose this to measure was because it's a very stable form of phosphorus in sediment. So we know that the isotopic signature are preserved well within this form. We calculated our theoretical equilibrium values using our contemporary monitoring, which sits on that bar in the figure on the right, as you can see. And what it shows is that in the early part of the record, there are some periods where phosphorus was not fully utilised with values lower than equilibrium. But with the introduction of the natural filtration system in 1991 that started to limit the amount of phosphorus coming in, we see equilibrium values where phosphorus was a limiting nutrient. Essentially, this was doing what it was supposed to do. But if we then look at the top part of this record, what we see is an increase in our phosphate oxygen isotope values above equilibrium. And this was a little bit confusing, and I'm working through this data at the moment. This is unpublished, and we are working to get it published now. But what we think is happening here is that if the phosphorus was in excess, we know that the phosphate oxygen isotope value should be closer to their original source value, which was 15 per mil, as shown in the earlier figure. To get values that are above equilibrium, we must have some sort of fractionation process occurring to result in these values. And actually what we found is that we know some cyanobacteria, particularly that microcystis that I mentioned, they use different phosphorus transport systems to enable them to take up and store phosphorus within their cells, which gives them a competitive advantage when they're under a P-limited environment. And when they use these different phosphorus transport systems, there is a positive fractionation effect that causes this sort of shift above equilibrium values. This is a disequilibrium process. So we're no longer exchanging phosphorus with the water because they're storing the light phosphate oxygen in their cells. Essentially, what this study has proven is that this increase in phosphate oxygen isotope values is a result of cyanobacteria undertaking different types of phosphate transport to adapt and thrive in a high nitrogen but P-limited environment, which has resulted from those changes in sewage treatment management over time. That was a major whistle-stop tour of stable isotope biogeochemistry. I'm hoping it was helpful for everyone. But I think the key conclusions here is that stable isotopes are a really powerful tool across all sorts of different disciplines and studies. We can evaluate both sources and processes across a large range of proxies and archives. And in particular, they have value in contemporary pollution studies where we can link together sort of nutrient biogeochemistry and the biological responses in both modern and historical contexts. And that is essential knowledge for our environmental management moving forward. Thank you very much. I've got a QR code here on my email if anyone has any questions or would like to get in touch, and we may have time for questions now.
Brilliant. Thank you so much, Savannah. That was genuinely excellent. That was a really good reminder of all of those isotopic systems. So now we are open for questions. So the way we normally do it is you type your question in the chat, or you just put your hand up and ask a question. So I'll give everyone a minute to formulate those if anyone has any burning questions at the moment. Lots of really positive chat. Lots of thank yous and positive claps, etc., popping into the chat here, Savannah, if you can see. Does anyone have any questions? Okay, well, I have one while people are still thinking. Oh no, we've got one from Nancy. If you'd like, you can unmute, or I can read it out, whichever you prefer.
Okay. Oh, yeah. Great talk. I love the topic. So my question was, since I'm doing carbon and nitrogen isotopes, are we able to like distinguish between abiotic and biotic sources in the environment?
Yes, definitely. So combined carbon and nitrogen, those cross plots that I showed that had the different sources. Obviously, I focus quite heavily on the organic sources, but it is very possible to find terrestrial source, like sort of non-organic sources as well. So the best way to do that is to combine your isotope data as well with the carbon to nitrogen ratio. So that was in that table as well. That's a really clear distinguisher for abiotic sources of carbon and nitrogen because it is quite different. The ratio of carbon to nitrogen is quite different.
Cool. Thank you.
Excellent. We've got another question from John. John, feel free to unmute or I can call it out.
Yeah, thanks. Excellent talk. I was just wondering, you mentioned that dissolved oxygen has been used to estimate rates of respiration and photosynthesis. I was just curious how that works.
It's an excellent question. It's not something I'm actually aware. I don't know the answer to it, unfortunately. It's not something I've done myself. I know that obviously the primary way that oxygen isotopes is used is hydrological cycle related.
Yeah.
I think that you can also measure the oxygen isotopes within CO2, for example. So it's possible that maybe that is a factor within it, but it's not something I'm totally familiar with, I'm afraid.
Okay, thanks.
Do we have any other questions? Not at the moment. So what are you doing now, Savannah? What are you currently working on? Obviously, you presented quite a lot of case studies there, but what else are you working on?
So aiming to publish the phosphate oxygen stable isotope core record, that's sort of what I've been working on quite heavily. But what I've also been doing is the nitrate isotopes in the waters. So I haven't presented it here because I haven't quite got it together, but much like those studies from those Chinese lakes, I've been doing the same principles within my own study sites, and trying to look at whether we can trace that sewage nitrogen signature through a catchment, and in particular pairing that with phosphate oxygen in water to look at whether it's P limited or N limited through a catchment scale.
Yeah, that's really fascinating. I think given the current status of some of our waterways at the moment, particularly thinking like Lough Neagh in Northern Ireland and many other catchments in the country, that it is a particularly challenging time for water quality and a really important thing to be managing and tracing that. So I think if we don't have any other questions, we will call it there. That was perfectly timed. Thank you very, very much. So I just want to say a massive thank you from MinSoc to you. As you know, these talks are recorded and not everyone's always able to make it on the day, but we do get loads of people watching the videos. And I really feel like your introduction was such a good grounding in isotopic systems that it's going to be really well watched. So once again, a final thank you. And we look forward to chatting to you again and good luck with the rest of your work.
Thank you very much, everyone, for coming.
Cheers. Thank you. Bye now.
Bye.